This lecture continues our series of 3 about microbes of the rumen. Here we'll discuss bacteria. As you recall from the last lecture, after these 3 lectures you should be able to identify each group of rumen microbes and their predominant species, explain their unique characteristics, and delineate their role in fermentation. And here we'll focus on the rumen bacteria. As we mentioned last time, the bacteria have activity towards many different substrates in feed. In fact, they're usually classified or grouped according to the substrates that they use. Table 9-1 in the Russell textbook groups the species according to the substrates that they use. And as I have indicated here, you need to know this table in some detail. The table lists predominant species of cultured bacteria and their niches, their substrates. It also lists the products they produce. That is less important for our purposes. What's important to know are the species and their niches, and the remainder of the lecture will cover this in some detail. The cellulose-degrading bacteria, or the cellulytics, attack cellulose, a polymer of beta-1,4-linked D-glucose in the cell wall, and you can see the structure of cellulose in the figure at the bottom. There are 3 predominant cultured species: Rumenococcus albus, Rumenococcus flavifaciens, and Fibrobacter succinogenes. There are more minor cultured species like Butavibrio fibrisolvens, Eubacterium cellulosolvens, and Clostridium species. These are considered more minor either because their primary niche is not degrading cellulose or they're simply less abundant in the rumen. The Rumenococcus species are gram-positive cocci. Rumenococcus albus forms white colonies. It produces acetate, formate, ethanol, and hydrogen. Rumenococcus flavifaciens produces yellow colonies, succinate, formate, and acetate. The reason for pointing out the color of the colonies and what products they produce is this is how we distinguish these 2 species, as you recall in an earlier lecture. It may not be inherently important that Rumenococcus albus forms white colonies and Flavifaciens produces yellow colonies, but this is one of the ways that we distinguish them as separate species. They can use pentoses as a source of energy, but they cannot grow on them alone, and so they are rather specialized on cellulose. Fibrobacter succinogenes is a gram-negative rod. This produces succinate in pure culture, hence the name. They cannot use pentoses, and so they are even more specialized than the Rumenococcus species. They use cellulose, and that is really their only niche. Butavibrio fibrisolvens is a motile gram-positive rod. It is weakly cellulytic. Some strains are completely non-cellulytic, and most strains lose their cellulytic activity shortly after they're cultured. This species is versatile. It uses many other substrates, and cellulose degradation is not its primary niche. We'll see that on subsequent slides. The Clostridium species include some cultured species, but these are not very abundant. Uncultured Clostridia are likely important. Sequencing has identified many bacteria that are firmly attached to feed particles as Clostridia. We've inferred from their attachment that they degrade cellulose or some other component of the cell wall. The cellulytic species have unique growth requirements. They grow under a pH of 6 to 7, hence the reason why fiber digestion is depressed if rumen pH stays below 6. They require or benefit from several growth factors, including ammonia, branched-chain VFA, phenolic acids, and unknown growth factors. They use ammonia for amino acid synthesis, and for a long time it was thought that they used only ammonia and they were not able to use amino acids directly. The branched-chain VFA are used for branched-chain amino acid synthesis. The VFA and their corresponding amino acids are shown below. For example, isovaleric acid is used to synthesize leucine. The phenolic acids are used to form the cellulosome, which, as we'll discuss later, is used to degrade cellulose. It's the enzymatic complex responsible for bacterial attachment to cellulose and the degradation of that cellulose. Phenolic acetic acid and phenylpropionic acid are used for cellulosome formation. There are other unknown growth factors that cellulytics require. Some species cannot grow in the absence of rumen fluid. This indicates that there must be some factors, we don't know what they are, that are required for growth. We call them growth factors exactly because we don't know what they are. Otherwise, we would give them a more specific name. The hemicellulose and pectin-degrading bacteria attack the other components of the cell wall. Hemicellulose is a heterogeneous polymer of xylose and other pentoses, and we can see xylan, which is an example of hemicellulose, down at the bottom. Pectin is a heterogeneous polymer of D-galacturonic acid and other sugars. These are the other components of the cell wall. The predominant hemicellulose and pectin-degrading bacteria are the Prevotella species, Butyrvibrio fibrosolvens, and Lachnospira multipara. The Prevotella species are gram-negative rods. They include Prevotella ruminicola, Prevotella brevis, Prevotella bryanti, and Prevotella albensis. These are the most abundant bacteria in the rumen. They make up about 30% of the total, and they are versatile. This is not their only niche. Lachnospira multipara is a gram-negative curved rod. It is motile, and it's flagellated. It degrades pectin but not hemicelluloses. It also uses sugars. The starch-degrading bacteria attack the storage polymer of alpha-1,4 and alpha-1,6 linked glucose that we know as starch. It is a non-structural carbohydrate. There are several species that participate in the degradation: Ruminobacter ammophilus, Succinimonas amylolytica, Selenomonas ruminantium, Streptococcus bovis, Succinivibrio dextrinisolvens, and Prevotella species. Ruminobacter ammophilus is a gram-negative rod. It has the highest activity and abundance of starch-degrading bacteria. We named a number of starch-degrading bacteria, but this is the most important species. Succinimonas amylolytica has lower activity and abundance versus Ruminobacter ammophilus. Similar niche, it's just less important because it has lower activity and abundance. Selenomonas ruminantium is a crescent-shaped flagellated bacterium. This is one of my favorite just because of its appearance. They look like little moons. Only some strains degrade starch. It is more important as a sugar and a lactate utilizer, as we'll discuss later. Streptococcus bovis is a gram-positive streptococcus. It can degrade starch, but it's more important as a sugar utilizer. We call it Streptococcus bovis just through force of habit. Its valid name is actually Streptococcus equinus, according to Berge's manual, which is the reference for this sort of matter. The reason for the change is that Streptococcus equinus and Streptococcus bovis were originally described as separate species, but Berge's manual later made the determination that they should be the same species. Streptococcus equinus was described earlier, so its name supplants Streptococcus bovis as the valid name for the species. Rumen microbiologists still call it Streptococcus bovis, and I doubt they'll ever adapt to the change. Succinivibrio dextrinisolvens is a gram-negative curved rod. It degrades maltodextrins, which are breakdown products of starch. They're starch oligosaccharides. It is not highly abundant. Protein-degrading bacteria are those that break down protein to peptides and amino acids. They may also ferment peptides and amino acids, but their activity towards protein is much greater. The diagram at the bottom helps illustrate this principle. The wide arrow connecting protein and peptides indicates that these bacteria have high activity towards breaking down protein. The thin lines through the rest of the figure indicate that it has low activity towards fermenting peptides and amino acids. These bacteria are found in high concentrations at 10 to the 9th per milliliter or higher. They are still saccharolytic. Saccharolytic means that they degrade carbohydrates. That is to say that degrading protein is not their only niche. The predominant species include Prevotella, Selenomonas ruminantium, and Streptococcus bovis, and we've seen that these are important for degrading carbohydrates. The amino acid degrading bacteria are named somewhat similar to the protein-degrading bacteria, but their activity is quite different. They ferment amino acids and peptides, and they do it 10 to 20 times faster than the protein-degrading bacteria. They do not break down protein. So we can see in the diagram below with the red X that they do not break down protein to peptides, but the wide arrows throughout the rest of the figure shows that they have high activity towards peptides and amino acids. They have very low concentrations, only 10 to the 7th per milliliter. That is to say, they're outnumbered by the protein-degrading bacteria by 100-fold. But because they ferment amino acids and peptides at a faster rate than the protein-degrading bacteria, these are still important for amino acid and peptide fermentation, despite their low concentration. They are asaccharitic. That is to say that they do not use carbohydrates. Their niche is degrading amino acids and peptides. That's all they do. The predominant species include Streptococcus bovis, Clostridium aminophyllum, and Clostridium sticklandi. These species were not discovered in the rumen until the 1980s, when Jim Russell tried to grow peptide and amino acid degrading bacteria. They weren't discovered because their abundance is so low compared to other bacteria. They had to be grown on selective media. Besides their high activity towards amino acids and peptides, one thing of note is that they are sensitive to the ionophore and feed additive monensin. The sugar-utilizing bacteria use glucose, fructose, xylose, and other monosaccharides, which are normally present in low concentrations in the rumen, but can be there nonetheless. The predominant species include Streptococcus bovis, Selenomonas rumenantium, Lactospira multipara, Eubacterium rumenantium, and Butyrvibrio fibrosolvens. We already talked about Streptococcus bovis, but I want to revisit it and talk about its role in acidosis. This species is voracious, and it can cause the rumen to spiral into acidosis when the animal is fed high grain, particularly when the animal is unadapted to the high grain. This species has a very fast doubling time. It's less than 20 minutes with high carbohydrate. In the lab, we've measured doubling times as low as about 15 minutes. You can almost watch it grow in front of you. It produces lactate, and lactate is highly acidic. It has a pKa of less than 4, which means rumen pH tanks when lactate accumulates. Streptococcus bovis is acid tolerant. It can grow at pH 4.5, whereas most bacteria die when the pH is below 5. So in some, Streptococcus bovis can grow quickly, it produces lactate to exclude other bacteria, and itself is acid tolerant, so it can grow under the conditions that it's created in the rumen. Normally, Strep bovis is found in low numbers in cattle-fed forage or adapted to grain, but again, when the animal is unadapted to grain, Streptococcus bovis can proliferate. Eubacterium rumenantium is a weakly gram-positive, non-motile short rod. All strains ferment sugars. Others can ferment maltodextrins and other carbohydrates. It's not well studied. It hasn't received much study in the last 30 to 40 years. There are several utilizers of short-chain fatty acids in the rumen. Short-chain fatty acids can arise either from feed, such as silage, or end products from other bacteria. Lactate utilizers include Megasphaera elsdeni, Selenomonas rumenantium, and Antivibrio lipolytica. Wollanella succinogenes can use malate and fumarate. These are never found in the rumen in high concentrations, but regardless, there is a bacterium that can use them. Megasphaera elsdeni is a gram-negative coccus. It accounts for up to 80% of lactate turnover in grain-fed animals. And the question I pose to you is, how can it save the rumen from Streptococcus bovis? I'll leave it up to you to answer the question. Go back a few slides, review the material on Streptococcus bovis, and then answer for yourself how Megasphaera elsdeni can be the savior of the rumen. We already talked about Selenomonas rumenantium, but we'll note here that it produces lactate and grows rapidly when sugars are abundant. It utilizes lactate otherwise, so it can be an important lactate utilizer, but its role depends on rumen conditions. Some people will unconditionally call it a lactate utilizer, and that's not really true. As we mentioned, Wollanella succinogenes can use malate and fumarate. Specifically, it makes succinate from hydrogen as well as malate and fumarate. It is not particularly abundant because it must compete with methanogens for hydrogen. It's been used mostly as a model, as a hydrogen utilizer in experiments. Its contribution to rumen ecology is probably very minor. Anaerobia lipolytica is a gram-positive rod. It utilizes lactate, but it's more important in fermentation of glycerol, as we'll discuss soon. Lipolytic bacteria are those that can hydrolyze triglycerides to fatty acids and glycerol. They ferment the glycerol, but they cannot ferment fatty acids. Fermentation of fatty acids is thermodynamically unfavorable due to the high hydrogen in the rumen. You may hear several explanations for why bacteria do not ferment fatty acids in the rumen, but this is the root cause. It's simply thermodynamically unfavorable with high hydrogen. In digesters with lower hydrogen, fermentation of fatty acids can occur. The predominant species of lipolytic bacteria is Anaerobia lipolytica. Biohydrogenation refers to an unsaturated fatty acid being reduced to a saturated one, and we see that in the figure below where an unsaturated double bond is being converted to a saturated bond. We'll follow Russell's textbook, and we'll say that the predominant species is Butyrvibrio fibrosolvens. We'll discuss and qualify this statement later in the semester. There are other bacteria that don't appear in Table 9-1 of the Russell textbook, but appear in the text itself, and these are important. We'll mention them here. Lactobacilli species are gram-positive rods. They ferment sugar and pectin. They produce lactate. These are more acid-tolerant than Strep bovis, and they can displace Strep bovis during the severe and last stages of acidosis. We oftentimes mention Strep bovis exclusively when talking about the onset of lactic acidosis, but this species plays a critical role as well in the terminal step. One last group of species discussed in the Russell textbook are the spirochetes. These have a unique morphology, and I wish I had a better image of them, but we don't grow them in the lab, so I was not able to take an image of them personally. These are gram-negative, and they have their unique spirochete morphology. It's known that they ferment sugars, pectin, and other carbohydrates, but otherwise they're not very well studied. In the rumen, there are both generalists and specialists. Generalists are species like Butyrvibrio fibrosolvens that utilize cellulose, hemicellulose, starch, pectin, and sugars, are the Prevotella species which use starch, hemicellulose, pectin, and protein. We've discussed these bacteria in piecemeal. For example, we mentioned that Prevotella uses hemicellulose in one slide, and it's only in another slide that we mentioned that they use protein. But don't let the broader concept escape you that some bacteria are capable of using multiple substrates. There are other bacteria that are specialists, like Fibrobacter succinogenes, which uses cellulose, and Ruminobacter ammophilus, which uses starch. These are the only substrates that these bacteria use. They are specialists. Another point to consider is that bacteria participate in cross-feeding. The product of one bacterium can be the substrate of another, and we already saw one example of cross-feeding in talking about the short-chain fatty acid utilizers. To paraphrase a literary source, no bacterium is an island. Bacteria exist in a web of interactions that we see diagrammed below. We will talk more about this point later. It's not important that you know the figure. Just keep in mind that bacteria interact with each other with cross-feeding. And one last caveat. Most bacteria, 90% or so, have not been cultured. This is suggested by 16S rRNA gene sequencing. When we take a sample of rumen fluid, we extract DNA, and we sequence the 16S rRNA gene directly. Only about 10% of the sequences match those of cultured bacteria. At best, cultured bacteria can be considered models. For example, we can say the ruminococci and Fibrobacter succinogenes are not the only cellulitic bacteria in the rumen, but they're probably representative and are good models for studying uncultured bacteria. At worst, we're missing 90% of the important actors. We'll discuss more of this later. The take-home messages for this lecture are that there are about 200 cultured species of bacteria. 20 to 30 species are predominant. It may seem like we discussed an endless list of bacteria in lecture, but compared to the 200 cultured total, we have discussed relatively few. These bacteria collectively occupy diverse niches. Some bacteria individually are generalists, and some individually are specialists. Lastly, uncultured bacteria are outnumbered by cultured ones several-fold. We study the cultured bacteria in depth because that's what we know, but our picture of rumen fermentation could be missing many pieces. This lecture